Hello, I'm Charles Cantor, and I'm speaking to you from the beautiful Polish city of Gdansk, where I'm here to help inaugurate a new European framework program on genomics and proteomics. And today I'm going to talk to you about genomics. I'm going to focus mostly on cancer, and the thrust of my topic is how to discover and use non-invasive biomarkers to monitor the status of a tumor or the status of therapy. So we like to use nucleic acids as biomarkers because they perform really well in a clinical diagnostic setting. And we like to use these non-invasively for reasons that are pretty obvious. So our choices to get samples of nucleic acids non-invasively are somewhat restricted: blood, urine, a few other places. But blood is best for two reasons. Blood communicates with almost every distant site in the body. So in principle, if events happen, they may release molecules that can be seen in the blood. And we have a tremendously well-developed infrastructure to obtain blood from patients, distribute it, purify samples from it, and so on. So let's think about, in a sample of blood, where might DNA come from? Well, almost all the DNA is inside nucleated blood cells. Once those are set aside, what remains is very, very dilute. So if there are cells from distant places, tumor cells, fetal cells in pregnancy, then there'll be DNA in those cells. But typically, there's about 1 or 2 foreign cells per cc of blood, compared with 100,000 or 1 million nucleated cells. So the cellular components of DNA in blood from distant sites are really too dilute to use. In addition to cellular DNA, there's about 10 times as much vesicular DNA, but that means 10 molecules, and that's still too little to work with. Fortunately, there's free DNA that's 10 times more abundant still, and typically free DNA from distant sites might represent about 100 molecules of any genomic sequence. So it's a very tiny amount, but it's enough to work with. In a typical situation, the host contributes a background of about 10 times as many DNA molecules. So you can see we're going to have 2 challenges. We have to detect very few molecules, and we have significant background. In the past, when people tried to isolate cell-free DNA from plasma, they didn't realize that this DNA was so rare, and as it turns out, that it's very low molecular weight. And they didn't realize that it was essential to avoid the breakage of host blood cells, which will drown out your signal with background. But we've learned a lot in the past few years, and that enables the kinds of studies that I'm going to show you. So to motivate this, let me challenge you to think about a funny question. What's the common feature in 3 very different clinical situations: Alzheimer's disease, cancer, and pregnancy? That common feature is cell death. So in all those 3 situations, and many other clinical situations, there is programmed cell death, and during apoptosis, cellular DNA is cut into nucleosome-sized pieces. So that's about 150 base pairs. And these pieces then enter the circulation. If the pieces from a distant site, a tumor, a fetus, a dying brain, if the pieces have different DNA molecules than the DNA molecules that are found in the white blood cell background, then by looking at these differences, we can infer the properties of a distant site without touching it. In the case of pregnancy and cancer, there are sequence differences between the fetus and the mother, between the tumor and the host. In other situations, like neurodegenerative disease or cardiovascular disease, there are no sequence differences between brain cells or heart cells and blood cells, but there are epigenetic differences. Okay, so by looking at either sequence or epigenetics, we can learn properties of a distant site without touching it. The challenge is that we have so few molecules. This leads to a fundamental physical problem called stochastic noise. If on average you have very few molecules and you take a sample, then because of statistical fluctuations, it's impossible to, in a single sample, get a precise representation of the true abundance. And this stochastic noise scales as the square root of the average number of molecules. So if I have 100 molecules, my standard deviation will be plus or minus 10. And if I use a range of 3 standard deviations for significance, that's plus or minus 30, which means I have almost no ability to quantify. If I have 5 molecules or 10 molecules, obviously I can't quantify, and I might miss a sequence altogether, so-called allele dropout. Okay? So this is a fundamental problem, and we need to solve this problem to work with cell-free DNA in plasma. And there are basically 2 strategies for trying to overcome stochastic noise. The standard approach is to repeat the measurements. But our measurements require 10 cc's of blood. And we consume the entire sample to detect our target. So it's not possible to sample 100 times. You can't take a liter of blood from a patient. So we need a different strategy. We need to find a large set of different but biologically equivalent markers, different DNA sequences, so that instead of averaging from sample from aliquot to aliquot, in a single measurement, we can combine the results from multiple sequences. And you'll see how we do that. Okay, now in trying to look at cell-free DNA, basically you need a platform that's very sensitive. And among the currently available platforms, 3 are widely used. You can do PCR, you can do DNA mass spectrometry, which is essentially PCR detected by mass spectrometry, or you can DNA sequences, DNA sequencing, second generation DNA sequencing. And the choice among these platforms is determined by cost, by error rates, but honestly, mostly by throughput. So if a single locus sufficed, you can use PCR. But in the problems I'm going to show you, a single locus does not suffice, either because we're trying to suppress stochastic noise, or because we have to measure many loci at once to be sure we've captured clinically relevant information. Okay? And so we'll use either DNA mass spectrometry or DNA sequencing. And I'm going to show you next non-invasive prenatal diagnostics, which uses DNA sequencing. And then I'll show you non-invasive cancer diagnostics, which currently uses mostly DNA mass spectrometry. Okay. Non-invasive prenatal testing has been commercially available for 2 years now. This year alone in the US, between Sequenome and other companies offering commercial tests, we will have studied at least 300,000 women looking for common fetal aneuploidies. We only look at the high-risk population, and in the US there are 750,000 high-risk pregnancies each year. So in the second year of clinical adoption, we will have tested almost half of the available market, and it's said that this is the fastest uptake of a new clinical procedure since the invention of penicillin. So what I'm going to do is to show you the principles and then show you some results of this procedure. Oops, sorry. Okay, so we're using peripheral blood. We take a 10 cc draw. That yields 4 cc's of plasma. And we're going to analyze that plasma DNA by random shotgun sequencing, as you'll see. And the process is actually pretty simple. But there's a problem. In an average pregnant woman, 13% of the DNA comes from the fetus, and 87% comes from the mother. But in individual pregnant women, there's a range of a factor of 20. And if a mother has only a few percent fetal DNA, there's too much stochastic noise to make the measurements. So we have to have a way independently of guaranteeing that there is sufficient fetal DNA to get above a statistical threshold. And in the commercial tests that we provide, that threshold is currently 4%. If we fall below that threshold, essentially the fetus becomes invisible. And so in a pregnancy, when we test, we will conclude that the fetus is female because the mother's female, and we'll conclude that the fetus is normal because the mother is normal. Okay? So that's not good. If the fetus is actually a male, we can use Y chromosomal sequences to ensure that there's enough fetal DNA in the sample. But of course, half of all fetuses are female, so we need a different approach. And for those fetuses, we currently use epigenetics. So we searched the whole genome and found sites which consistently are methylated in the fetus and unmethylated in the mother. These are imprinted genes which are silenced during fetal development by methylation. And we looked for sites that had that property, and in addition, they fell in sequences that are recognized by restriction enzymes that are inhibited by methylation. So what we do is we take the mixture of fetal and maternal DNA, as shown here. So we start with a mixture of fetal and maternal DNA, and we cleave it with a cocktail of restriction enzymes. And what happens is that the fetal DNA remains intact, but the maternal DNA is cleaved. And we now have only pure fetal DNA. So when we quantify that, it tells us the number of fetal molecules. So here's what that looks like for one imprinted locus, one methylation-sensitive locus. In women who are not pregnant, there are no molecules of methylated DNA detected. Even earlier than 8 weeks in a pregnant woman, we see methylated sequences. And after 8 weeks, when we would actually perform non-invasive prenatal diagnostics, we have a nice, robust signal. But 2 cautions. You see, it's tremendously different from patient to patient. And here the average number of fetal-specific sequences in our sample is only 140. So we're definitely in the domain of stochastic noise. All right, so I'm going to show you a cartoon that illustrates the principle of how we quantify the abundance of chromosomes in the mother's blood. Here I've color-coded the DNA fragments so that the red ones are from the fetus and the blue ones are from the mother. And okay, so this is an average pregnancy. So 13% of the fragments are from the fetus. What we're going to do is sequence starting basically from single molecules, amplified, and then read. We read 36 bases from one end of these fragments. And if the read matches a human genome assembly, we can assign that fragment to a chromosome. If the read doesn't match, we discard it. And in practice, we make about 12 million reads. And this allows us to build up a histogram of the abundance of each of the different chromosomes in the mother's blood. A large chromosome, like chromosome 1, will generate a lot of fragments. A small chromosome will generate very few. And we know from independent experiments that the whole genome, both maternal and fetal, are represented more or less evenly in the mother's blood. However, if the fetus is aneuploid, so for example, if it has an extra copy of a chromosome, then the fetal contribution from that chromosome will lead to an imbalance in counts. So if the fetus has 3 copies of chromosome 21, there'll be extra counts from chromosome 21. It's a small effect because if the fetal DNA is only 10% of the whole, and we're going from 2 copies of chromosome 21 to 3, then it's a 5% effect. So to be able to measure that very confidently in the presence of stochastic noise takes this rather elaborate procedure. But let me show you what that looks like. First, I'm going to show you results from that clinical trial that led to the launch of this test commercially. So this trial was managed by Women's and Infants Hospital in Rhode Island. It involved about 2,000 patients, 27 centers worldwide. All of these patients were women who had high-risk pregnancies that had volunteered to undergo invasive testing, either amniocentesis or chorionic villus sampling. And before the invasive test, we took our 10 cc's of blood. We then looked at the relative abundance of chromosomes, and what I'm plotting here are results for the 3 most common aneuploidies, trisomy 18, 13, and 21. And what I'm showing on the vertical scale is a statistical measure of whether the representation is normal or abnormal. And what I'm plotting horizontally is the percentage of fetal DNA. And what you can see is that some of these pregnancies obviously have an overrepresentation of chromosome 21 or 13 or 18. I want you to notice that we don't look at women with less than 4% fetal DNA, and that in general, there's a pretty good correlation between the amount of fetal DNA and the strength of the statistical deviation, exactly as there should be. Okay. So since these women all had invasive tests subsequent to the blood draw, we could use the results of the invasive test as a gold standard and compare the non-invasive test and we could basically get a pretty good picture of how accurate we were. And this summarizes the results on the common trisomies and also on the X and Y chromosome aneuploidies, determination of gender if someone wants that, and also results from twins and triplets. And the bottom line is that the results are not perfect, but they're close to that. The error rates are very, very tiny. The results are far better than 99% accurate. Okay. So this was a research project, a clinical trial. Subsequently, we transferred this test into our CLIA lab, our clinical diagnostic service lab, and they refined it. They improved the sample preparation. They improved the methods of analyzing the data. They tweaked the sequencing procedures. And today, the results are so good that it no longer makes sense to use conventional invasive testing as the gold standard, because, in fact, the sequencing is more accurate. So this shows results for chromosome 21 on about 100,000 pregnancies. It's the same kind of plot, the statistics of statistical significance of a deviation from normality. Here you can see a very, very tight Gaussian distribution of that statistical parameter for normal pregnancies and a large number of trisomies. So this test works extremely well, but again, it is not perfect. If we look over here at very low fetal fractions, although our cutoff is 4%, we'll make an occasional error if it's a pregnancy with 5 or 6%. We could raise the cutoff, but then there'd be too many samples that are uninformative. With a 4% cutoff, we can actually make a definitive call on 99% of the samples, which is extremely good. Okay, so we get false negatives in samples which don't have enough fetal DNA. We get false positives for a variety of interesting biological reasons. There's a phenomenon called confined placental mosaicism, in which the fetus is normal, but the placenta is trisomic. And since most of the circulating DNA from the fetus actually originates from the fetal portion of the placenta, we'll make a miscall. Another example is what's called the vanishing twin. You start with a twin pregnancy. One is normal, one is aneuploid. The aneuploid twin dies. This is quite common. And then you have a placenta that represents a aneuploid twin, but the other twin that remains is normal. So we make errors like that, and occasionally we see other things that at first were confusing. So here is results for a pregnancy, very high fraction of fetal DNA, it appeared. It looked like a positive z-score means that you have an extra copy of a chromosome, and a negative z-score means that you are missing a chromosome. And it looked like this fetus was had an extra copy of 21, was missing 13 and 18. In fact, this has very little to do with the fetus. This pregnant woman also had metastatic carcinoma, and we were sampling DNA from the tumor as well as the fetus. Here's another case. In this case, it looked like 21, 13 were deficient, 18 was present in excess. These results were so surprising, we repeated them, and as you can see, the replicate is excellent. But once again, this is not the pregnancy. This pregnant woman also has an invasive angiosarcoma. Okay, so you'll see in a few minutes when we transition to cancer that it's the same principle, just not as mature. We also see other things. at the current sequencing depth, if there are subchromosomal but large insertions or deletions, we see them. Here, for example, is the abundance of counts across most of the genome. And you can see that just part of the short arm of chromosome 18 is elevated. Well, when we see something like this, we don't know if it's the mother or the fetus, unless it's a known syndrome, and the mother is clear of that syndrome. We can assess that by going back and looking at maternal Buffy code. But this leads to a very amusing and interesting situation, because we see in roughly 1% of all these pregnancies, we see chromosome abnormalities that in some cases are well understood, like Angelman syndrome, and in other cases are just mysteries. In fact, if we're willing to sequence more deeply, we can see anything and everything. And 3 years ago, with Dennis Lowe, we sequenced the entire genome of an unborn fetus at horrendous expense, admittedly, but it can be done. 2 years later, several groups reproduced these results at much less cost. And in a few years, it's probably going to become practical to do this on every pregnancy, if you wish. But suffice it to say, this raises some pretty horrendous ethical issues. You may not want to know the whole fetal genome sequence of your unborn child. Okay, well, we're going to switch now, more in line with the subject of this meeting, from non-invasive prenatal diagnostics to non-invasive cancer diagnostics. And we're going to use mass spectrometry for that for several reasons. In cancer, we are mostly interested in looking at genome abnormalities that drive the properties of the tumor. So cancers have unstable genomes. They have a mutator phenotype. So the genome is going to hell. And there are millions of abnormalities in the genome of a cancer cell. But most of that is noise. What we're really interested in predominantly is somatic mutations that lead to the aggressiveness and the particular properties of a given tumor. And the number of those somatic mutations is finite, and it can be managed still by DNA mass spectrometry. The advantage of DNA mass spectrometry over sequencing is throughput. We can measure hundreds of samples a day with one instrument instead of just a few if we had to do DNA sequencing. So you may not be familiar with DNA mass spectrometry, and I want to just briefly show you what the properties of this tool are. First, let me show you the context in which we're currently trying to use it. Oops, wait a minute, sorry. Okay. We want to focus on somatic mutations that make tumors sensitive or resistant to drugs. For a given patient, we'll normally be able to start with a biopsy, but then we want to transition from a biopsy to measurements in plasma, and just fixing a little glitch on the screen. Thank you. Okay. Okay. So DNA mass spectrometry, we use PCR to amplify simultaneously many loci in a single microtubule well. We have to do this because we want to look at many potential oncogene mutations simultaneously. And once again, we have to consume the whole sample to make the measurements. The PCR amplicons are too long to fly in a mass spectrometer, so we subdivide these by a subsequent sequencing reaction. With DNA mass spectrometry, we only look at intact DNA molecules. The samples are not allowed to fragment in the mass spectrometer. And so if I have a sample of DNA after PCR, it consists of only 4 bases. and they have very different molecular weights. So we can predict from the sequence the expected molecular weight of any DNA fragment, and that means that we can design assays in software, and they'll work, and it means that we can analyze mass spectra from lookup tables. So this is very, very simple. Just to make sure you see what the process is, here's a schematic picture for a single biallelic locus. If it's heterozygous, when we do PCR, we make two amplicons that differ in sequence by a single nucleotide. And when we then sample this sequence difference by placing a sequencing primer next to it and extend by a single base, we'll make this blue fragment that ends in G by copying the C allele. And we'll make the green fragment that ends in C by copying the G allele. And when we fly just the sequencing products in the mass spectrometer, we'll see two peaks. And if they're equally abundant, they'll have equal area. And in fact, the area of these peaks represents concentration with a precision of 1 or 2%. So this is a very accurate way of detecting not just sequence, but also abundance. But we do this not just on one locus. We do this on many loci all at once, 10, 20, 40. So we know currently about 700 somatic mutations that define the properties of cancer. And depending on the tissue and type of the cancer, our knowledge of important mutations ranges from very complete to barely scratching the surface. And I'll show you an example from breast cancer because it's kind of average of our current understanding. So this is a study done using DNA mass spectrometry at MD Anderson Hospital and published last year. They were looking at women with breast cancer. These are invasive measurements, fine needle aspirates, and they looked at 267 samples with methods that had a sensitivity that was probably around 5%, okay? And at that sensitivity, they could see, having tested probably 500 mutations, they could see 37 different mutations in this cohort. And 40% of the women in this cohort had one or more of these somatic oncogene mutations, of which the most common are in PIK3CA. Now, what's amazing is that every one of these mutations is clinically actionable. So for this lucky 40% of breast cancer patients that have one or more of these mutations, it's possible to choose an optimal therapy. But this is still invasive. And so now we want to go from invasive to non-invasive. And to do that, we have to raise the sensitivity. So in a typical biopsy of a solid tumor, a somatic mutation can present at a few percent or up to 50%, let's say, because a biopsy of a tumor is really a mixture of normal cells and tumor cells. Now, when a tumor cell dies, its DNA is diluted by normal cell-free DNA in the host. And so instead of needing a sensitivity of 5 or 10%, we need a sensitivity that's much higher. And I'm going to show you that, the kind of method that's used to achieve that higher sensitivity. So our problem in detecting, let's say, a 1% mutation or a 0.5% mutation in a background of a vast excess of wild type is that when we perform our sequencing reaction, the wild type template consumes the sequencing primer, and we may not be left with enough sequencing primer to detect. So to solve this problem, what we do is we omit the substrate that it allows the sequencing primer to extend on the wild type template. We add only substrate that allows the sequencing primer to extend on the mutant template, and therefore we essentially eliminate the wild type peak from the mass spectrum, and now we only see the mutant peak. And if we watch that peak in a set of spiked samples, you can see that even at less than 1% mutant, we see a very distinct peak in the mass spectrum that identifies the mutant. We call this technique SABER, S-A-B-E-R, and this version of it is a little tricky, but in a few minutes I'll show you another version that's more robust. Let me pause just to say that I notice people are sending in questions, and I'll deal with the questions at the end of the presentation, but I don't want to interrupt to do it right now. Okay, so, oops. All right, so here is a study from Japan in which they used this very sensitive SABER procedure to monitor tumor-specific DNA in plasma. The patients are lung cancer patients, and these patients were chosen because they had activating mutations in EGFR, a tyrosine kinase, and they were being treated with tyrosine kinase inhibitors. Now, the problem with using these very effective but specific drugs is that the mutative phenotype of the cancer provokes the creation of mutants which are less sensitive or insensitive to the drug. And with tyrosine kinase inhibitors of EGFR, the most common resistance mutation is T790M. This does not eliminate the utility of the drug, but it compromises its effectiveness. So this group looked at about 70 patients being treated with tyrosine kinase inhibitors. And if you look just on the left of this slide, the green curve represents patients that did not have this compromising mutation. And the red curve shows patients that do have the T790M mutation. And you can see that the prognosis of the patients with the T790M mutation is considerably worse. So this is useful. It's non-invasive, so you can employ it over and over again. But it's a very limited study. Next, I want to show you results from Johan de Bono's laboratory in London. These results did not use the SABER tool. They just used conventional DNA mass spectrometry. So their sensitivity is less, 5 or 10%. But it's a much bigger study. These patients are all phase 1 clinical trial patients. They have very advanced disease. They have a large tumor burden. And as you'll see, many of them have abundant cancer-specific DNA in their plasma. So this slide compares the average DNA concentration that we see in pregnant women with what de Bono sees in healthy volunteers and his late-stage cancer patients. And you can see that when he compared healthy volunteers and late-stage cancer patients, there's about a factor of 8 elevation in total DNA in plasma. This suggests, but does not prove, that most of that DNA is tumor-specific. Before I leave this slide, let me just mention we don't think the difference between pregnant women and healthy volunteers is significant because the methods used to quantify the total DNA were quite different. But now let's focus in on this difference that de Bono sees between healthy volunteers and cancer patients. So this breaks it down, DNA abundance, total DNA abundance in blood, in colorectal cancer, breast, etc., all the way to healthy volunteers. And you can see that in almost every kind of cancer, there's significantly more DNA in blood than in healthy volunteers. You can also see from patient to patient a huge range in DNA concentration. Again, this is total DNA concentration. Now, if you combine all these results, you can ask the question, does the total DNA concentration in blood, is that a parameter with any clinical value? And it turns out that it is very valuable. So patients with low total DNA blood concentrations have a very good prognosis, and patients with high DNA concentrations in plasma have a very poor prognosis. And since this is just total DNA in blood, these measurements could have been made in 1960 if people knew not to break the host cells and knew to capture these small DNA fragments. And these results have now been repeated in other sites. So in addition to just looking at the total DNA concentration, we can ask, if we have a biopsy and plasma from the same patient, do we see the same somatic mutations? And the answer thus far is about half the time. And in half a dozen other studies that have been published in the past year, it's always the same answer, roughly half the time. And there are three reasons for this. Most of the methods in use are not sensitive enough. And so in tumor samples or plasma samples, then with less than a few % tumor-specific DNA, those signals are lost. Second, stochastic noise. In patients who have very few molecules, then there's allele dropout. But finally, there's a biological situation here. Remember that in plasma, we're looking at DNA from cells that have died recently, because plasma DNA has a half-life of about 15 minutes. Whereas in a sample of the tumor, we're looking at mostly tumor cells that are alive. So it's not surprising that there's a lot of discordance. If we use more sensitive methods, then we can eliminate the problems of missing low abundance alleles. But we still will have to deal with stochastic noise, and we'll have to deal with biology. But this clearly argues that to push the field further along, we need methods that are a lot more sensitive. And I'm going to show you a method that we're about to launch commercially, which I think answers this need. So it uses the Saber principle, just as before. We don't provide a substrate that allows the wild-type sequence to be read. So the only sequence that's read is the mutant sequence. But we supply that substrate with a capture tag, a biotin, so that after primer extension, we can capture the mutant sequences with streptavidin-coated magnetic beads. And then in the mass spectrum, there's no wild-type allele. There's only a mutant allele. And this approach can be multiplexed, so we can look at many mutations at the same time. So here's what this looks like for some spike-in samples at 1%, 0.5%, 0% mutant. The red dots are capture controls to prove there's DNA there. And you can see that we have no trouble at all detecting 0.5%. And there clearly is very little background. So this method has a sensitivity that's close to 1 part per thousand. And think of it, if you have 1,000 molecules and only one of them is a tumor, specific sequence, you couldn't use a method with a sensitivity any higher even if you had it, because there wouldn't be a molecule to detect. Well, this shows a clinical sample in which it's actually a archival paraffin embedded sample, in which there's an activating EGFR mutant present at a high abundance, and in one sample, a very tiny amount of mutant T790M EGFR sequence, and you can see that it's quite visible. So this looks like a very good tool. We've practiced it now on 8,000 different mutants, and this truth table, the correct answers in blue, the incorrect answers in red, show that while not perfect, this method is tremendously good and is very robust, very easy to use. Okay, so we have a tool that can be employed on plasma of cancer patients, and this will be commercially available, we expect, by the end of the year, and it covers 26 different activating oncogene mutations. 24 of those are shown here, and those of you familiar with some of these mutations will realize that this is going to be useful for sure for colon cancer, lung cancer, some breast cancer, and melanoma, and so on. So this is where we're at. Now, this is detection. But as I said at the beginning of the talk, we're working in the domain where we can't quantify. And that's a pity, as I'm about to show you. So if we could quantify, then we would have a way of assessing the clonality of tumors. And we can do this on biopsies where there are enough molecules so that quantification is possible. So if I have a biopsy, it's a mixture of normal cells and tumor cells. And if all the tumor cells are identical, and they have more than one driver somatic mutation, then those mutations should occur at the same allele frequency, because the mutation frequency is just the percentage of tumor cells. And let's test this on a series of lung cancer samples. So patient number 88, this is results from Japan. 2 different driver mutations, same allele frequency, looks like a clonal tumor. Patient 81, again, 2 different driver mutations, but same allele frequency, clonal tumor. Patient 25, this mutation is present in most of the cells, these 2 in very few cells, clearly not clonal. Here's another case. Patient 471, 2 driver mutations, same allele frequency, clonal tumor. Patient 407, 2 driver mutations, very different allele frequencies, not clonal. So why this is important, if you have a clonal cancer, you're dealing with one disease. And it's possible that a monotherapy would be effective. But if you have a non-clonal tumor, it's very unlikely that you'll be able to monitor the patient effectively. You'll be able to treat the patient effectively with just a single drug. Okay, now, so it's frustrating at the present time. We can detect things, but we can't quantify things because of stochastic noise. And to be able to quantify things, we have to use the trick that I described at the beginning of the talk. We have to aggregate results from multiple markers, and there just are unlikely to be enough informative somatic mutations to do this. So we need another approach. And that approach is to use epigenetics, just as we used it in pregnancy to make sure that we had enough fetal DNA, we had to make a quantitative measurement. So we can also use methylated sequences from that are tumor specific, if we have enough of them to form a signature that will be statistically useful. And this paper that I'm going to summarize was published 2 years ago, and it used DNA mass spectrometry to look at methylation after bisulfite treatment in breast cancer patients. And let me show you first just this one marker. This is one of the BRCA genes. This is a sample of normal breast tissue, and you can see that this marker is mostly unmethylated. There's some methylation, but it's low. In a pure sample of tumor, this locus is almost entirely methylated. And in blood from this patient, it's almost entirely methylated. So this tells you that this patient has breast cancer, and it tells you that most of the DNA in the blood of this particular patient comes from the tumor. But they had not just one, but quite a few different markers. Here is a sort of heat map from 8 markers on normals and tumor carrying women. And again, these are blood measurements. And so you can see there's pretty good contrast, and clearly you will gain in statistical confidence if you combine results from multiple markers. There are various ways of doing that. And in this work, they used principal component analysis. So here are the first several principal components of the differences in methylation between red breast cancer patients and blue normals. Again, these are measurements in blood. And you can see, while it's not perfect, there are a few outliers. There's pretty good discrimination. And this is not limited to only 8 markers. One could combine many more. And it's not limited to pure methylation alone. We could combine methylation and somatic mutations. So to summarize, looking at plasma DNA in pregnant women is a very successful commercial diagnostic test being performed on hundreds of thousands of women a year now. Looking at the same type of plasma-specific markers in cancer patients is just beginning. It's a research tool, but I think we can be pretty optimistic that we'll begin to see widespread use in the clinic within the next few years. And with that, I want to thank you for being such a patient audience. And let's see if I can handle the questions by clicking on this button. Okay, so the first question has to do with skin biopsies of leukemic cancer patients. It's a subject I've never heard about before, and I'm really not sure if it's useful. But I would consider any biopsy to be invasive, and the question was, is this method invasive? It might be useful, but I know nothing about it, so let me just say, don't know. Okay, how do we do this? Let's see if there are more questions. Uh-oh. The questions is always the hardest part of this. Answer is missing. Okay. No questions available for display. Well, I'll give you a couple of minutes to ask me some questions. We'll see if any questions come in. No? Oh, do you usually get enough cell-free DNA for sequencing? Can I use whole genome amplification to generate cell-free DNA for my downstream sequencing? Okay, so here, I don't understand this, but I don't like whole genome amplification because it distorts quantitative features. So you can do it, but I recommend against it. Okay, we're still waiting for any more questions. Okay, I guess maybe there are no more questions. No, this is blinking. Okay, let's see again. Okay, so I'm being asked, the idea is to replace biopsies in the follow-up. And the answer is absolutely yes. That's what we want to do. Yeah, no follow-up biopsies. This is something strange about this. Okay, so the question is, how reliable is this? What is the evidence? And the answer is too early to tell, but preliminary results look quite encouraging. Okay, is this going to become the standard of care, is the question, and the answer is yes, I think it's going to become the standard of care. There have been almost 10 publications this year from all over the world showing proof of concept, and now we just need to refine the methods and do studies on large enough numbers of patients to have statistical significance. Okay, I think I'd like to thank you. I think we're going to, I don't see any more questions. And I just remind you that this lecture is eligible for CME credit. So if you want those, please look at this slide. And that's how you arrange to get credit.